Hello everyone, let's get started. Today's video will involve our virtual lactase lab exercise. I'm on Canvas right now, and this is the view of the week 10 module, the beginning of our online classes. And where it says for Tuesday, March 24th, Lactase Lab Day 1, go ahead and click. That brings us to the link for the Lactase Lab Day 1 handout. Go ahead and click that as well. So this is the handout that we'll be essentially going through together today, again in a virtual version of our laboratory exercise. What I'd like you to do is pause the video for a moment, download the handout on your computer, and save it, because there will come a point in the video where you'll need to answer some questions directly on this handout. Now, the last time we were together, prior to spring break, we were talking about enzymes. We talked about how enzymes are catalytic proteins and how they are necessary in all living cells because they help cells perform the metabolic reactions of everyday life. We talked about the three functions, specific functions that enzymes perform in living cells. We talked about how enzymes bring substrates in close contact with each other at the active site. We talked about how they lower the activation energy of the reaction, which allows the reaction to hit that important transition state more readily. And we talked about how the enzyme and the substrate go through a shape change in order to achieve that. And finally, we talked about how enzymes, through their functioning, increase the rate of the reaction and therefore increase the rate of product formation for the cell. If you recall, we also talked about how enzymes have a different effect depending upon environmental conditions. Remember, we said that for every organism, the internal enzymes are adapted for that particular organism's environment. For example, in humans, our enzymes function optimally at a pH of about 7.3 and a temperature of 98.6 approximately. Remember, we said that that's the optimal conditions for functioning, and that enzymes are capable of functioning at lower temperatures and higher temperatures within a particular range, lower pH and higher pH within a particular range, but that there is one optimal temperature and one optimal pH for particular enzymes in every organism. In today's exercise, we're going to be using an enzyme called lactase. You can identify this term as describing an enzyme because the name ends in ASE. It's going to be important to remember that carbohydrate names end in OSE, because today we'll be using a carbohydrate called lactose and an enzyme called lactase, and it's easy to get those two terms confused. The lactase enzyme that we'll be using for our experiment actually comes from a bacterium called E. coli. And the optimal environmental conditions for this particular version of lactase are pH of 7.3 and a temperature of 22 centigrade, which is about room temperature. Now, what we're going to do in the experiment is first run a control to see how this enzyme behaves at optimal pH and optimal temperature. We're essentially going to feed the enzyme increasing amounts of substrate, and we're going to check and measure to see how much product the enzyme makes. Then we're going to change the pH and change the temperature so that we can see what those effects have on the ability of the enzyme to make product. Remember, when you change pH, what you're doing is you're changing the amount of free hydrogen ion in the solution. And depending upon how much free hydrogen ion there is, some of that hydrogen ion will be binding to and interacting with the enzyme. Remember, any time that you put something onto a protein, you stick something onto a protein, you're going to cause that protein to change shape. And even a slight change in the shape of a protein will change its function. So by altering pH today, we may or may not be altering the shape of this lactase enzyme and impacting its ability to make product. The two pHs that we're going to investigate are a pH of 5 and a pH of 8. So an acidic pH and a more basic pH. We're also going to alter temperature today. We're going to look at a 0 degree centigrade and a 50 degree centigrade. And we're going to see how that affects lactase ability to make product. Remember that temperature has a very different effect on enzymes. Temperature is about kinetic energy. And as we change temperature, what's essentially happening is we are changing how often the enzyme interacts with the substrate and therefore how readily it can make product. If you increase the temperature, you're going to increase the kinetic energy of the molecules in the reaction tube. That's going to result in perhaps more frequent collisions between substrate and active site, but only to a point. When we get to a high enough temperature, we're actually going to cause a degradation of the enzyme. The enzyme is actually going to denature or essentially lose its tertiary and even secondary structure. So it'll be interesting to see whether or not 50 degrees centigrade is hot enough to do that. On the other end, at low temperatures, we're slowing everything down inside the test tube, and we're making it a little bit less frequent, a little bit less often that the substrate and the active site of the enzyme collide. So we may see that there is less product made at these lower temperatures. Remember that at a point, the enzyme will be cold enough that we may stop seeing product formation altogether. We will have to wait and see whether or not a 0 degree temperature is cold enough to stop product formation. Now, I just want to review for us these two important molecules that we're working with today, lactase and lactose. Milk is the source of our lactose sugar, and lactose can be found in all different kinds of milk that comes from mammals anyway. Lactose is actually a disaccharide. It's a molecule that's composed of two simple sugars bonded together. Those sugars are galactose and glucose. These sugars are joined together with a BIO110 glycosidic linkage. What do you remember about BIO110 glycosidic linkages? Remember when we talked about carbohydrates and we said that there are alpha linkages and there are beta linkages. Can you remember the difference between them in terms of how easily or how difficult it is for an enzyme to break them down? When milk is consumed, the lactose sugar gets broken down by the lactase enzyme into its two simple sugars, galactose and glucose. And that's what we're looking at down here in this drawing and this essentially a description of this reaction. The other figure you have in your handout is of the brush border cells of the small intestine. Remember, these are the cells that line the intestine, and these are the cells that would be secreting this enzyme and then absorbing the products of the reaction, the glucose and the galactose. Before we left for spring break, I had mentioned to you that there was a little bit of a snag in this experiment because we're not able to measure the products of the lactase reaction directly. If you recall, I told you that we have to use an alternative substrate today. This is a molecule that has a long and complex name. And now you can see it on your handout, but know that we refer to this substrate as ONPG, O-nitrophenyl beta-D galactoperoxide. It's quite a name. ONPG actually looks a lot like lactose, and it's capable of binding to the active site of the enzyme lactase. Now, it doesn't make the same product molecules because it's not lactose, but if you look at the reaction down here in figure 3, you can see that ONPG will be broken down by lactase to make the simple sugar galactose and another molecule called ONP. The nice thing about ONP is that it has a color. And so depending on how much ONP product is in the test tube, you will get a higher and higher absorbance with a spectrophotometer. So what we'll be doing today is we'll be feeding the enzyme lactase the ONPG substrate, and we will be measuring the ONP product that gets formed. So here we have the goals of our experiment. Number one, we just wanted to determine the relationship between the amount of substrate that we feed this enzyme and how much product it can make. We're actually going to feed the enzyme increasing amounts of ONPG, and then we're going to see how much product it makes over a set period of time. Now that's an important thing to notice about our protocol. We're not going to determine the rate of product formation. We're simply going to look at the overall trend of how much product is formed depending upon how much substrate we give to the enzyme in the test tube. The second goal for the experiment is to look at the changes that occur when we alter the environmental conditions and basically repeat the experiment. Put different amounts of ONPG substrate into the tube with the lactase enzyme and see how much product gets made at a particular interval of time. We'll then be able to compare the control reaction at a pH 7.3 and 22 centigrade or room temp, and when we alter the pH or we alter the temperature. And what we'll do is we will look at a data set together and we will make some graphs from that data set so that we can see exactly what happens to our enzyme's function when we alter these environmental parameters. So we'll be using a spectrophotometer to measure the ONP product that is made in our chemical reaction. This is a very similar protocol to the one that you performed when we did our protein assay at the beginning of the semester. And if you recall, the more protein that was in that tube, the higher the absorbance reading on the spectrophotometer. We'll see that exact same thing today, with the difference being that we will actually be making the product in the tube because we will be putting an enzyme in. All right, that's it for this first video. I want you to go ahead and stop this video now and open up the next one, and we'll continue on with the experiment.